So thank you for introduction. So good morning, everyone. So I'm Honda Naoki from Nagoya University, Graduate School of Medicine. So first of all, I'd like to thank the organizer for bringing me here, such a great seminar, and especially Nakai-san. He's my collaborator, actually, and he has been kind of crazy friend for many years. So today I'm going to talk about kind of machine learning approaches to uncover the genetic role of the brain-wide wiring via SPERRFY. SPERRFY is the name of the method we develop as a machine learning method. Okay. It's really a pleasure to share my recent work. Next slide. So actually, this work was mainly conducted by Koike-kun, Jigen Koike, who is my PhD student right now. So he's in the Hiroshima University student. The Hiroshima University is my former university. I recently moved to the Hiroshima to Nagoya, actually. Then now he's now studying in Nagoya at a place, as a place. But anyway, so and also this guy, including Nakai-san, technically support his work. And also this work was already open in the preprint server by archive. Okay, then let me introduce my lab before going to start. So I'm a PI of the data-driven biology lab at Nagoya University. And our lab works at the kind of intersection between the machine learning and the mathematical modeling. And we apply this approach to a wide range of the problem in the biology field. So as you can see that we studied a really broad range of the topics, and our lab is organized into 4 groups actually: the neuroscience decision-making group, and the omics analysis group, and pathological modeling group, and the computational modeling group, kind of like that. And today I will focus on one of these omics-related group, which connect directly to the theme of this talk. Anyway. Okay, so actually I'm really interested in the design principle of how neural circuits are developed during development. Then despite many anatomical and, where's my pointer? Oh, sorry, I will find pointer. Where's my pointer? Oh, yeah, I got it. Yeah, I'm really interested in design principles in circuit development. Then so many study, examine the kind of Brodmann map or functional map and the connectivity, but we still do not fully understand this fundamental question: what rules shape the wiring of the entire brains? So this question is a core of today's talk. And when we think about what shapes neural circuit development, there are 2 major forces. One is experience. For example, the somasensory and the visual and the auditory input can reorganize the neural circuit through the activity-dependent plasticity, right? So probably, and on the other hand, on the other side, we have a genetically programmed mechanism, including the development process and the molecular gradient that guide the action during the development. Of course, both of these are very important, but today's talk I will focusing on the genetic rule of the wiring, this side, okay? So to understand how neural circuits develop, so we need to bridge the hierarchical gap between the microscopic and macroscopic level, okay? At the level of the cellular level, which is microscopic, so axon guidance regulated by molecular gradient distributed in the brain tissue. Okay, developing neuron. During development, the neuron extends several processes called neurite. Okay, this is the neurite. Then the one with neurite rapidly elongated to become an axon. Whereas others neurite are going to be dendrites. The tip of the axon here which is called Grosscom. Okay, so it can show the chemotactic ability and migrate in response to the attractive and repulsive gradient Q. Okay, and finally reach and make the connection with the target neuron. So that's the cellular and the molecular process of the axon guidance. In contrast, at the circuit level, okay, the brain exhibits highly organized large-scale wiring pattern. Okay, as you can see, like a diffusion tractography. So this gap leads to the fundamental question. So how do local molecular rules scale up to the global, brain-wide wiring? So that's our question. Okay, and so as I mentioned, the axon guidance is regulated by the gradient of the attractive and the repulsive cue. That over the years, so many family of the guidance cue and their receptor has been identified, such as efferring, the neutering, and the semaphoring, which are very famous axon guidance cue manual. So in this sense, we already have a rich knowledge of the molecular basis of the neural circuit formation. But despite knowing so many molecules, we still don't fully understand how these Q collectively, regulate the wiring at the level of the whole brain. Okay, that's our question. And in addition to the molecular mechanism, as I just mentioned, so here I'd like to introduce the idea of the information representation in the neural circuit formation. Okay, which is based on the Roger Sperry's chemoaffinity theory. Okay, so he's a kind of main player in this talk. But anyway, so Roger Sperry is very famous, widely known as a Nobel winner, winner of the physiology of medicine, and for his work on the differentiation of the cerebral hemisphere, left-right hemispheres. But he did another great contribution, which is chemoaffinity theory. So he studied the visual system consisting of the retina and the tectum. Tectum is a kind of visual area of the brain. Then the retinal ganglion cell here extend their axon to the tectum region, while preserving their spatial relationship like this. Okay, there's such a circuit called, such a mapping or such a projection is called topographic map. Okay, this is topographic map. So that's why the spatial image, reflected on the retina, is transmitted to the visual area tectum like a camera. Okay. The SPERRFY, the Sperry proposed that both source area and the target area, there is a molecular gradient. Okay, so in this case, the FA receptor gradient, efferring ligand gradient, and also and interesting orthogonal direction, the efferring big ligand gradient is in the retina, and the FB receptor gradient in the tectum. Okay, that's interesting. The axon, so each axon projected to the location where molecular concentration match, like this. Okay. So this matching mechanism generates orderly topographic projection. So in other words, the molecular gradient provides positional information that guides the matching between the source and the target region. Okay, so that's really important concept in my talk today. And here in this slide, I briefly mentioned two of my previous work on the mathematical modeling for the neural circuit formation. So actually, we developed a molecular level model to describe how axon switch attraction and repulsion, attractive and repulsive response in response to the gradient. And also, build a model to explain the information representation as seen in the chemoaffinity theory. Our hypothesis is that mathematical modeling essence of is that axon, like growth cone, so, actively switch the attraction repulsion depending on the absolute concentration of the guidance cube. So this property, based on this property, topographic map is beautifully developed. And these models explain the very simple topographic circuit. However, these models do not generalize the entire brain actually. When we look at the real whole brain connectivity like this complexity is far beyond what classical model can capture. Okay. So it's difficult to address this complexity by mathematical models. So as you can see here, the cortical wiring forms an extremely dense and highly interconnected structure, right? So, but my model is very small, kind of toy model. So there is a hierarchical gap. So that's why. In the front of this reality, I feel this is hopeless to model the entire neural circuitry. So that's why we switch the mind. And I ask the question: is it possible to model the wiring? This hopeless situation motivated us to take different approaches, which is the data-driven approach.
So I have a question.
Okay.
In the original Sperry theory is global, so this kind of whole of the brain network or more local through.
Okay, that's a very good question. Actually, Roger Sperry, this chemoaffinity theory can be applied to the mainly sensory system. For example, the visual system and auditory system or somasensory system. Probably, this sensory system can be explained by chemoaffinity theory. But within the cortex, for the cortical interaction, we cannot. There's no case to apply the chemoaffinity theory. This is because cortical interaction is so complex.
Next question is that your previous two research depends on the Sperry theories or independent from the Sperry theory.
Okay. Our study is based on the Sperry theory, but this study is only for this question in this study is how Sperry's chemoaffinity theory can be realized based on the intracellular reaction network as a chemotactic system. So we want to understand in this previous study, we want to understand the molecular mechanism to realize this kind of the chemoaffinity theory. But in this talk, we will extend focus to the entire brain.
Okay, thank you very much.
Okay. As I say, these and especially my mathematical modeling is very limited to address the very complex brain reality. So actually, this limitation comes from this one, I guess. So actually, mathematical model works very well only when the phenomena has a very clear regularity. For example, like this. And also phenomena shows a distinct pattern. But biological reality is very complex and messy. So, for example, the many parameters exist, and these values are completely unknown. And of course, these systems are very high-dimensional systems, many variables. So that's why classical model, especially my previous models, mathematical model, cannot capture the brain-wide widening rule. Okay. Okay, in response to these challenges, the large-scale connectome project has been launched around the world, as you know, in the recent year. And they span the multi-species from human diffusion tomography and the mouse virus-based tracing and electron microscope-based connectome in the fruit fly, for example. These projects, of course, I have to mention in Japan, the Brain Mind project addresses the marmoset, common marmoset, common marmoset connectome, right? Especially Shimogori sensei, I guess. These projects provide us the high-dimensional dataset describing the brain wiring. And given such a dataset, what we want to understand is that the design principle of the neural circuit, I mean the wiring rule that guide the action during the brain development. Okay. So could the entire neural connectome also be wired according to the molecular gradient? So this is my question. I think key hint come from the work of the Shimogori sensei, Professor Shimogori from the Riken CBS. This is Shimogori's work, by the way. Here is a brain tissue. And we have several gradient of the transcription factor. If you apply the ectopic, the additional FGF gradient here, surprisingly, the cortical area, for example, the motor cortex, small sensory cortex, auditory cortex, visual cortex, these cortex areas can be duplicated. That's surprising. So this resembles the very classical French flag model famous in the development biology. Okay, so in the French flag model, so this is a coordinate of the tissue, and this is a gradient called this is the morphogen gradient. Then the local molecular Q, the absolute concentration specifies the specific area, in this case, the cortical area identity. Okay, but I guess this model is not enough to understand this duplication, because the cortical area function, emerge from its neural circuit, neural context, right? Because this is brain. The brain works within a network. That's why this function can be emerged embedded in the network. So this function is cannot be determined by solely local molecular concentration. Okay. So that's why we need to extend the concept, I guess. Okay. So I think neural circuit may also be organized by positional gradient as seen, but not only the level of the area differentiation, like classical developed biology, but also the level of the axon targeting, I guess. That's why, even complex intracortical circuit may follow the molecular gradient as seen in the topographic map, as seen in the chemoaffinity theory. Okay.
Okay, so I have a question. So Shimogami-san work, so in this case, how the size of the region, so whole brain or more restricted regions in this instance?
Your question is the size of the duplicated area. I think, actually, I didn't know exactly, but I guess, this area is small and because brain tissue is,
brain tissue, so this is...
Brain tissue is the same size, of course.
Okay, and so this is from a mouse cortex?
This is mouse brain,
Yeah, I understand. So that means that small cases, so there is so we can do it. And so your idea is a more general idea, so not only focus on local regions, so more global idea.
Global.
So your circuit idea is not local. So because there's a very long range connection. That means that your more general idea. So
Yes, thank you very much. That's right. And also, okay, so in this, what I want to say is that probably whole brain cortical connection also regulated by the gradient. Okay. Then, and also, but another question is there topographic organization in the cortical, circuit? And I found the what? Well, I found the classical illustration of the axonal fiber in the brain. So this is just a human sketch, anatomist sketch. So I don't know who is that, but anatomist, very probably anatomist, yeah, in this experiment, anatomist dissect and gently separate the long range cortical axonal fiber. But this is bundled fiber, and find that these fiber maintain their, this fiber projected to the specific region while maintaining their spatial relationship, as you can see. So as this is as in a topographic manner. Okay. In other words, topographic organization already exists in the long range cortical axon. Okay. As seen in the retinotecton visual system. So this kind of sketch, observation or sketch, suggests that spatial order is a fundamental principle of the cortical wiring, I guess. And this, very ancient old sketches motivated us to ask whether molecular rule, wiring rule may underlay this organization. Okay. And another hint of cortical, another evidence of the topographic organization in the cortex is the functional topographic organization. So this is a what? So this panel represents the orientation preference onto the visual cortex, V1, V2, V3, V4, blah, blah, blah. And this color represents the degree angle of the orientation preference. And you can see that these functional, identity is topographically, organized like this. And also similarly, auditory cortex, there is the similar pattern, similar topographic, functional pattern exists as known as a retinopathy. So these examples show that, even in the sensory cortex, have a highly topographic structure. And I think that this functional map, topographic map, may be supported by underlying the structural topographic, mapping, I guess. Okay, so here I want to introduce our key hypothesis behind the classical chemoaffinity theory. Okay, in the cortex, so this is the same cortex. So in the cortex, many defined molecular gradient, receptor gradient and ligand gradient are distributed like this, in a complex manner. So instead of the very simple one-to-one mapping like classical chemoaffinity theory, we assume many-to-many relationship. I mean, the axonal projection are guided by the combination of the multiple receptor and multiple receptor gradient and multiple ligand gradient. So according to such a multi relationship, I guess we can observe the defined type of the projection type. For example, the topographic type like this, that preserve the spatial over, and also this is topographic. Also, the convergent type. Okay, for example, the hippocampus, receives the many synaptic input from the variety of the brain area, right? This is a convergent type. And another type is divergent type. For example, the dopaminergic, VTA, the dopamine neuron in the VTA projected through various region of the brain. So this is divergent type. Okay, I think these, multi, many to many chemoaffinity theory explain such as these, projection type. But I guess so this data is very complex. That's why this is too complex to recognize the underlying the projection rules by just looking at such a high dimensional structure. So that's why we need a computational data-driven approach. And they given such a challenges. So we took a top-down approach, took top-down idea. So instead of trying to build a mathematical model, very toy mathematical model, so we think we can infer the latent wiring rule from with the use of the RM brain atlas in this study. So here, so RM brain atlas provide the two kind. So this atlas provide many modality data. But here we are focusing on the connectome data and the spatial transform data. Okay, in this transform data they provide where the neuron with specific location project to. So we know that kind of departure site, departure and destination site, right? Okay. And also in the spatial transform data, so more than 9,000 gene, as a gene expression of the more than 9,000 gene are, measured as by using the classical insight hybridization. Okay. By combining these two datasets, we can obtain the pair of the gene expression at the source site and the gene expression at the target site, right? From this paired vector data, we aim to identify the axonal projection rule in a data-driven manner. Okay. Then toward this end, so we developed the data-driven framework to discover the multi-chemoaffinity wiring rule. So this is called SPERRFY. SPERRFY. So with respect to the Roger Sperry, anyway. So name of formal name is kind of like this. Okay, then the SPERRFY, our method to SPERRFY integrated two data set. So there's a connectome data set and gene expression data set. And then by integrating these, so we repeat the same explanation anyway. So then we obtained a pair of the gene expression vector of target source and target site. And from these paired data, we want to infer the latent gradient that regulates the actual wiring rule. Okay. Which means our method in our method to SPERRFY extend Roger Sperry's concept to the whole brain. Okay. Then this is the basic idea of the SPERRFY framework. Okay. So here we have a source brain area and the target brain area. Okay. Let's imagine this is a two kind of the area. Then each area there is many gradients of the molecule A, B, C to molecule D, E, F, kind of like this. Okay. Okay, I got to mention that this is a very simplest case. So we assume for the simplicity, we assume the coordinate is one-dimensional for the simple explanation. Okay, this is one-dimensional area, one-dimensional target area. There is many gradients of different molecule, and then then we assume that there is a gradient which provide the positional information for the wiring. And this gradient is achieved by the weighted sum of these gradient. Okay. And this is also. So this is a gradient wiring positional information gradient. Okay. This is also, obtained by the weighted sum of the actual molecular gradient. Okay. Then we assume in this model, in this concept, we assume that axon located here, okay, here, project to the here with the same level of this gradient. Okay, these gradient completely match, right? Okay, of course, this axon here project to this one and this one. So this is our very simple mathematical model underlying the SPERRFY framework. Okay. So in the next step, I will explain how we actually estimate these weighted sum of weight using the CCA. Okay. For
So
this,
can I,
yeah.
so, right, so in this case, the source area and the target area is a different regions, the different region or the same regions.
Okay. Either would be okay actually. So for the simplicity, for simple explanation, I say these are different area. But in our study we address this is a whole brain, and this is also a whole brain.
And
Of course, because we address the cortex axonal projection.
yes, and the next question is you show the molecular A, B, C, and D, F. So this is different and the same actually.
Yeah,
So,
actually, it can be same. It can be same. Yeah, it's confusing for the audience anyway. So, yeah, A, B,
okay,
C, D. Yeah.
and you are also using RNA through data set, and in
Uh-huh.
this case, there is a sort of atlas of the sort of region of interest. So do you use these kind of sort of regions? So yeah, how do you sort of explain the sort of the brain itself? So more so surface level, more the detailed level sort of regions, or more so some kind of sort of 200 to 300 sort of separated regions in brain.
Yeah, actually, I skip the one model. Yeah, okay. So we address yeah, actually,
Um,
yeah, RM brain. Yeah, I forgot to cite the original paper anyway, or nature paper. Okay. Then this is a RM brain atlas mouse brain connectome data. Then they each, yeah, if you carefully look at, is a zoom up? I don't know. I zoom up this slide, but probably Zoom cannot
yeah, I
reflect.
can see. Yeah,
But
you.
anyway, so you can see that each point, right? Each point represents correspond to the virus injection site actually. then then they measure the axonal projection by based on the fluorescent signal. Anyway, so sorry. My answer is very bad. So Nakai-san's question is
Yeah, so you
Nakai-san question is what is a what
yeah, so you separate the
spatial
brain.
spatial resolution of the
Yeah, right.
row, right? Okay. So actually, yeah, I forget separated. Yeah, this is the RM brain atlas provides a ROI of the each brain region. For example, the isocortex and the olfactory area, blah blah blah. And then each major region can be also divided into the several ROI. In our analysis, we follow their definition of the very local ROI. But yeah, I don't know how to say.
Right. So you're divided 213 regions. You
Yeah,
like,
yeah. 213 regions. Yeah.
and this kind of simple explanation is so there's continuous so some kind of axis, but actually the 230 vectors.
Mhm. Yes, that's right. That's right.
Okay, thank you very much.
Yeah. Uh, yeah. Okay, so let's go back to this idea. So this is a mathematical idea behind the SPERRFY method. And then I explained these molecular gradient is weighted to sum, weighted and summed, and it become the positional information wiring the red gradient. These correspondence determine the axonal projection. Okay, then how to estimate the W? So actually, oops. This estimation was performed by the CCA, canonical correlation analysis. Uh, So, okay, then so in general, CCA is a method to extract the correlated structure between the paired vector x and y. Okay, x and y. In the CCA in general, there is a x vector. Each circle corresponds to each component of the vector. Okay, this is a y. This is also vector, and then we calculate the weighted sum of these components. So this is kind of the inner product of the between the x and a vector. So this is a u. Also, we calculate the weighted sum as a v. Okay. Then in the CCA, we optimize this weight a and b weight in order to maximize the correlation between u1 and v1. Okay, this is a CCA. And also, anyways, there's another inner product, weighted sum, U2 and V2. This is also, this correlation also maximized by tuning the A2 and B2 vector. But I have to mention that the U1, A1, A1, A2, A3, A4, these are orthogonal, orthogonal restriction. Then, so CCA is kind of like this. As you can see that, so in the CCA, U and V are we assume these are highly correlated, right? In our SPERRFY framework, this value, if you focus on each connection, this value and this value is the same, right? This value is the same. This value same. Which means these values are super highly correlated, we assume, in this model. So that's why, by estimating the W, for estimation of the W, CCA is very suited for this estimation. Okay. Okay. Then if you apply so how CCA works? In the case of the very simple retinal tactile visual system. Okay. As I explained that in the retina and the tectum, there is a F' gradient in the orthogonal direction, F'B and FB gradient distributed like this. And if you apply this CCA to this case, I guess U1 and V1 is distributed like this. And then V2U2 is distributed like this. Okay. So that's why, so if you apply CCA into this simple case, so we can automatically extract the latent gradient. Very obvious, trivial latent gradient like this. Okay, then here we apply now. let me explain how we apply the SPERRFY to the real bounce brain connectome data. So this is a connectome data. Then this is a connection matrix, okay? Then you can see that there is a several major region, okay? Very big ROI, major region. Each major region contains the several ROI, small ROI. In response to Nakai-san's question, so there is a more than 200 ROI region on the right hemisphere. Okay. So this is a connection matrix. This is a binary matrix. Okay. Then totally we have more than 2,000 connections. And also RNA atlas also provide a gene expression atlas, special gene expression profile like this. Then here we use around 700 gene because we only focusing on the developmental related gene. So we only we narrow down the number of the gene to around 700. Okay. Then by combining these data, we can get the paired gene expression, data, source and target data. Then we apply the SPERRFY. We apply the CCA.
Sorry, so I have one question. So in your cases, so you only focus on the right hemisphere. So how do you think that the left to right is also sort of work on your theory, or why you focus on the only right? Yeah, because sort of right to the left, the wrong connection is also existing.
Actually, sorry, I forgot in detail. I forgot my student analysis menu. If my memory is correct, actually, real data injection site is not so many actually, and that's why to increase the data set, I think left and right hemisphere are integrated onto the right hemisphere. Okay. Do you understand what I mean?
Yeah, I don't understand. Only so injection is only right. Mostly is right.
Yeah.
Okay, thank you.
But
Yes, Honda-san, can I ask a question?
Yeah, sure.
Okay, yeah. So did you use the data in adult mice or developing mice?
Actually, so this is adult mice. So we are really interested in the development process, but in this study, we are only focusing on the adult mouse. So
Okay,
because
so then if the gene expression data in the developing stage becomes available, that is also very interesting.
I know. And the RNA brain atlas also provide a gene expression profile in the developing stage. But unfortunately, brain morphology is really different to the adult brain. So that's why we need
more
dedicated to registration or something like that. So that's why as a first step, I skip this process. Yeah.
Okay, great. Yeah, I understand.
Okay, then we apply the CCA to the adult brain, connectome and gene expression data. Then this is a result of the CCA. Then we extracted for example, the 5 component of the correlated structure. These axes correspond to this one. Okay, for example, this is a source site. This is a target site. So this is a positional information, first position information. This is of the source site. This is first position information of the target site. This is second component. This is the third, 4, 5. Then we focusing on the top 5 components like this. Then these, weighted sum. This is a weighted sum, the inner product. Highly correlated, not highly, mind you. So high correlated. So now we estimated the W. We estimated not W, A and A. So this weight, A1, A2, B1, B2. That's why then we have gene expression data. These are available. That's why we calculated U1, B1 from the gene expression data. Then this is a visualization of the weighted sum. Which we call wiring positional information gradient at source and the target region. Okay. Then from this profile, I can say that, for example, the red brain region prefer to extend the red brain region here. Okay. The blue region, neuron in the blue region prefer to extend the blue region here. Okay. So this is the first component. The second component kind of like this. Red brain region, neuron prefer to predict this brain red region. So kind of like this. But only each pair cannot determine the connection. So whole combination of how to say, so all condition that 英語はジェネレートされない。すべての条件が揃ったらコネクションされるって言いたいんですけど、sorry in Japanese。そうだから、for example, the brain, yeah.
まあまあ、日本語でもいいですけど。
Probably, so this matching should be satisfied for not exactly satisfied. Softly, it is matching softly somehow satisfied for each connection. Okay. And okay. And also, so this profile is the same as this one. And if you more closely look at the local PI gradient value across the major region, in the lower panel, shows for example, in the lower panel, in the first component, the PI gradient, or this is the same. PI gradient is highly correlated. I think this is reflected by the local, very proximate, very local connection, very proximity-based local wiring rule is reflected by this highly correlated, gradient. And also, but the more interesting one is that more interesting pattern is that here, this is third component. There's a blue scatter, right? This region corresponds to, sorry, it's difficult to look at. Blue scatter corresponds to the thalamus. Okay. These are highly distributed, and other brain region is almost the same value, almost 0. And interestingly, in the target sites, the isocortex value is shows a various value, but another brain region shows almost 0, which means that this pattern corresponds to the thalamus neuron projected to the isocortical area. So, which is, of course, this circuit, thalamus cortical circuit is very well known. That's why our method probably collectively, extracted to such a reality. Okay. And but this is just estimation, just our estimation. I don't know how we cannot reliable 信用できない。sorry. 英語が出てこないぞ。ごめんなさい。How to say 信用できないってなんていうの？
Unreliable じゃない。
Unreliable, unreliable. This is our estimation. Results could be, can be unreliable. That's why to take this, to uncertainty, we perform the holdout variation. So this is the connectome data. This is original data. Then we separated the training data and the test data. Then we train the weight matrix or weight matrix of the CCA. Then we apply the test data. With the test data, and then this is a correlation coefficient between this one. This is top 5 component, but actually there is a top 50 component because we perform the dimension reduction of the PCA onto the 50 dimension. Anyway, then we found that this red line is the original correlated structure, correlation coefficient. But if you perform the holdout test, so even in the holdout test, top 5 component, correlation are stably, robustly maintained. But after 6 components, so correlation coefficient is significantly dropped in the holdout test. So that's why we actually focusing on the top 5 component. Okay. And this structure, created structure is robustly maintained. That's why our estimated result can be reliable. And also, okay, now we identify the wiring PI for top 5 components like this. But the next question is, what is the genetic basis of this wiring PI, wiring positional information? Then to test this one, so we computed the cosine similarity between the identified wiring positional information and for each each gene expression. Then we found the cosine similarity distribution. Then we screened that highly correlated, highly similar gene expression. Then this is a top ranked gene for each component. This is sorry, it's difficult to. This is so small. Sorry, this is first component, second, third, fourth, 5 component. This is the source region. This is target region. Each line corresponds to the highly correlated gene. And then we found that so we have many genes which can be the candidate of the gene wiring PI gradient. Then notably, for example, in the second component, we screened out the FB6 and the FNB2. This is a FFrin FFrin pair, which known as a regulator of the topographic gradient. So our even used of the adult mouse data. So we successfully extracted the development related region of that gene, and also for fourth component, Lobo2. This is a very famous molecule for the slit lobe signaling of the axon guidance. So that's why. So our analysis on the adult brain successfully related to successfully extracted the brain development related gene. Okay. And also, so to our SPERRFY framework, to confirm the reliability of the SPERRFY framework, we next perform the reconstruction or prediction of the actual brain connectome from the wiring positional information gradient. Okay, so this is a wiring PI gradient, positional information gradient. So as I said, the blue region, the neuron in the blue region prefer to the blue region, right? Then of course, this neuron does not prefer the to project the red region. Of course, this is the first component. The second component also, the blue region, the neuron prefer to project to the blue region, of course, but hate to project to the red region. That's why this wiring this is a U is the wiring pi vector, and the V is also wiring pi vector at the target site. This norm, so we computed this norm. This is a similarity of the wiring pi for each connection. Okay. Then we binarize these value. Then we reconstruct the brain, connection matrix by using the various threshold. Then we achieve that. This is the ROC curve, and we confirm that our wiring pi gradient successfully reconstructed the actual brain connectome with the 0.88 AUC performance. Okay. So that's why I think this result is really important because here we successfully predict the brain connectome structure from the only from the gene expression data. That's why I think based on given this result, I can say, we can somehow extracted design principle of the neural connectome in a data-driven manner, I guess. Okay. And this is final slide. This is final slide of our result. So, and also to make our result much more reliable, we perform the statistical testing. Okay. So this is the original. So here I will explain what kind of statistical testing is performed. So this is the original connectome connection data. Then now we perform we assume that these connections are globally randomized. Okay, we assume globally randomized model. From this data, artificial randomized data, we perform the SPERRFY to analyze. Then and also I forget to mention we have another neural model. This is a locally randomized model. So these color represent the major region, isocortex and olfactory valve, or kind of like this, olfactory cortex. Then within the brain region, these connections are randomized. This is a locally randomized model. Then we found that correlated structure is completely disappear on the globally randomized data, which means that our original data, this original estimation is obtained by chance. But in the locally randomized data, so interestingly, the first and second component, the correlation with the first and second component are still maintained. Okay, but the third, fourth, five component is significantly dropped. Similarly, this is so we also compute the similarity of the original PI gradient, and artificial data estimated gradient. Then interestingly, first and second component in the local randomized data, so these similarities still maintained, but third, fourth, five component significantly dropped. So this data suggests that the local randomized model captures these statistical testing captures the global local hierarchy in the wiring PI structure. So that's interesting. There is two kind of the regulation of the axon guidance. So probably two type of the gradient, regulate the regulates not local, local wiring, but globally, major region level wiring, regulate measuring wiring. But and also the third, fourth, fifth component, these gradients regulate the local connection of the axon guidance. So that kind of hierarchical regulation is really interesting for me. But anyway, so this is a summary, final slide. So we developed the new machine, not new, it's a CCA-based machine learning method called SPERRFY, which is inspired by the Sperry's chemoaffinity theory to extract wiring position information gradient from the brain data. By applying the SPERRFY to the mouse connectome and the gene expression atlas from the RNA brain atlas, we identify the robust wiring PI gradient, and also we recover biologically meaningful molecule. We screen the biologically meaningful molecule, including FF pair or robust signaling pair or kind of like this. And also we successfully reconstruct the connectome structure only using spatial gene expression data with high performance 0.8 AUC. And finally we reveal the hierarchical global local wiring rule in the brain. So this work was mainly performed by the Jigen Koike-kun, and also Nakai-san, Hira-san, and Yada-san also really support his work. So thank you very much.